Plasma is categorized into thermal and non-thermal. So non-thermal we are going to use in the food applications. So I'm going to show you how to generate cold plasma, its working principle and the components. This is the cold plasma setup. There it is a step-up transformer. So actually why we need step-up transformer? So to break down the gas, we need higher voltages. So that is a step-up transformer. So it takes it helps in giving the higher voltages. Then this is the treatment chamber. We can see upper electrode and the lower electrode, and there is a dielectric. This is a multi-pin dielectric barrier kind of setup. So in this we are using electricity as a energy medium to generate cold plasma. So you want to keep the sample? And one question you may get, why to treat with cold plasma? So obvious question, right? So actually, let's say the major macromolecules in food are proteins, starch, and lipids. Let's say. So, but proteins are starch in their native form. They don't have a desired functionalities. Let's say we have a starch, so we want to make a jelly from the starch, but native starch has a poor viscosity, which we can't use for the preparation of the jelly. So in that case, we have to use some modification techniques which can enhance its pasting properties. So the talk of this GIAN course is sustainability. So this is a sustainable technology we are using because it won't use any chemicals or it don't produce any pollutants. So it's a sustainable and green technology. So this can be used as an alternative to the chemical modification techniques for any biological macromolecules to modify. So the purpose of, let's say this is a spirulina. So let's say it has around 100 grams of spirulina has around 59 grams of protein. So it's a protein rich content. So you want to develop something, a protein rich product. So, but it don't have, let's say you want to use these proteins for stabilizing emulsions, but it don't have a that emulsifying capacity to produce a stable emulsion. So what we do, we use cold plasma to modify its functional properties so that we can it can be used as a better emulsifier. Gas can come little closer. This is the treatment chamber. As I said, this is the upper electrode, this is the lower electrode, and I am keeping the sample in between. So it involves high voltage, so it's dangerous. So so before moving to that, we should know what are the parameters which affects the extent of modification or degree of modification. So the major parameters are voltage. So the concentration of reactive species which is generated during the cold plasma are directly proportional to the voltage. So more the voltage, it breaks down more and it gives a higher concentration of reactive species. If it's less concentration. And the treatment exposure time. These are the major parameters which affect the extent of modification. So actually now we are using for protein modification, but it actually has been used effectively as a disinfectant. So it is very effective against killing microbes, especially in seafood where the microbial load is very high. So it's they are highly perishable and they have very disease causing microorganisms. So we can treat those seafood to kill microorganisms also. But here we are using spirulina to enhance its functionalities. So first we have to switch on. Actually, you can't see the cold plasma in daylight. Maybe you have seen, maybe you can search in Google after going cold plasma. You can see cold plasma in different colors, sometimes in pink, sometimes in violet. So this is because why, when you are giving energy to molecules, obviously it goes to the higher energy state or excited state, but they are highly unstable at the excited state. So obviously they try to come to the ground state. So when they come to ground state, they release that energy in the form of a photon. It's called light. So that's why you see a light when cold plasma generates. That's what I'm saying. In case of lightning, you can see a blue color lightning. Okay, so it's very difficult to see cold plasma in the daytime because of the higher light outside, but you can smell the cold plasma because it generates reactive oxygen species. Let's say now we are using, this is ambient air system, so cold plasma can be generated in the vacuum also. So in vacuum is it, now we have to, in order to produce cold plasma, we have to give at least 15 kilovolts, but in the vacuum you can produce cold plasma at 1 kilovolt. So but the disadvantage with vacuum cold plasma is those vacuum chambers and vacuum pumps are expensive and very difficult to commercialize. So to address that limitation, so they improvise to this one to atmospheric pressure. But here the disadvantage is we have to give the higher voltages. So I have turned on the, this is the regulator, this is the voltage regulator, and here we can set the time. So I will increase slowly. At some point of time you can hear the hissing sound of cold plasma. So even though I am increasing the voltage, at some point of time, at some point it reaches that threshold and air starts to break down into cold plasma. You can hear that. Obviously here it starts at 15. Now you can hear sound, right? I'll keep at 14 kilovolt. Okay, if you want to see, you can come near. And if you want to smell cold plasma, but it's very strong odor outside. But if you come near, you can uh, smell cold plasma. They react to oxygen species. Make it sound from. Maybe strong odor outside. That's it. So, but normally you can, if you want to see, you can come at the night time and you can see the cold plasma. Okay, I'm turning off, and we have different versions of. Actually, there are different setups of cold plasma. This is a dielectric barrier. So they are microwave assisted cold plasma and radio frequency and plasma jets we have, and we have corona discharge. They have different. They are specifically designed for different applications. Let's say when you are using plasma jet, it can be mostly used for preparing plasma water, which can be used for liquid samples. But here we can't use for liquid samples. It's mostly for the solid samples. And over there, it's an indirect application of cold plasma. First, we discharge plasma into water. So what happens? So plasma react with water, and it creates a completely different mixture of reactive species. So that water we can use for the sterilization, and we can use for other modification purpose. The only advantage of that is, let's say in the air plasma, most of the reactive species generated are very short lived, maybe nanoseconds. So once you turned off the cold plasma, it's gone. But when you discharge it into water and it reacts with water and produce plasma activated water, which can be active for hours. So let's say the recent application of cold plasma is, we know that seafood, after harvesting, or what we do, we pack in it this thermocol box and we add ice and we transport it to the longer distances. Nowadays they are replacing that ice with the ice prepared from the plasma activated water, which have antimicrobial properties. So which can give us a more shelf life because it can retain the plasma species for the long time. So one more thing, I will give a little intuition about how it inactivates the microbes. So now we are taking atmospheric air. So actually, the thing about cold plasma is reactive species. This plasma chemistry is so complex because, for example, if you are using air as a feed gas, so air has oxygen, nitrogen, and it has some moisture, and it has so many inert gases. So it's very difficult to quantify how many reactive species are forming and what are the reactive species forming and how these reactive species are responsible for the changes. So that is the main reason till now cold plasma is not approved as a disinfector or a processing techniques by United Nations. Okay, so then this is a research question for you to address. So because there is no specific criteria, so how much concentration of reactive species are forming, they know what are the reactive species forming, but how much concentration, how exactly they are responsible for the changes or inactivation, till nobody knows and nobody able to quantify that. So that is the main reason it's still in the lab scale or lab level. And yeah, the concentration depends on the concentration, but feed gas also, for example, you are using inert gas normally. Let's say you are only using inert gas in a vacuum cold plasma. There we have to give a separately air. So we have only one inert gas. Let's say in that case, the concentration depends on the number of reactive species or ions formed based on the voltage given. So here we are using atmospheric air, which is very complex, and we are breaking it down and making it even more complex to understand our study. And one more thing, even if a 2% of gas is ionized also, we call it as a cold plasma. So it's not like that we have to ionize 100% of the gas. So 3% ionization may have a different concentration, 4% have a different concentration. So, and one more thing, it's very difficult to reproduce the results. Today I am giving, so today the moisture, maybe humidity in the air may be 60%. So if I am giving the same treatment tomorrow, the humidity may be 90%. So it can completely change the plasma chemistry and give it different results. So the reproducibility of the results are also a issue in case of a cold plasma, especially ambient air. So they bring this to make it cost effective because we can use a atmospheric air, but still it have a limitation. So I will explain mostly we in a this fish industry or a life this marine industry, we mostly deal with the microorganisms. Actually all these technologies which we borrow from the pure science or techniques only. This is just applied branch we are applying. So it was first used in the biomedical for wound healing and microbial inactivation. Now we are adopting for modification of food, proteins, starches, lipids. So I will just give a brief idea about how it inactivates microbes. Then I will give little intuition about how it modifies the proteins and the starches. First, let's say let's we take a ambient air. So it's very complex. So I will take an example of oxygen. So how it breaks down and how it helps in inactivation of microbes. So let's say we have given the voltage to a certain threshold. Then what happens? This oxygen molecule will be broken down into so many components. Let's say atomic oxygen, and this atomic oxygen will be again recombined with oxygen O2 and form ozone. And there is a moisture, and it reacts with that, and the H2, this moisture is H2O is broken into HO, hydroxyl oxide. Then it recombines with the atomic oxygen and it produces H2O2, hydrogen peroxide. So which is also a disinfectant. So these are the major species which are responsible for the inactivation of microorganisms which are produced in the plasma. Okay, we have a sample, let's say fish. So we kept. So what happens? These are the reactive species, oxygen species generated, and they are highly reactive and ready to react. So let's take a microbial cell. So what happens first? They react with the cell wall. So they actually the cell wall and they make a holes in the cell wall. So as soon as they make a hole, so the fluids inside the cell start to come out. and it creates a space for the other reactive species to enter inside and damage its DNA and it functional enzymes. Enzymes are also protein. So what happens? These proteins will unfold and lose their functionality. Maybe that microbe won't be dead at that time, but it lost its functionality. It can't replicate, and so maybe after 1 hour, 2 hours, so it can't replicate, and microbes will be dead. So this is first they break the cell wall. So then they modify the proteins and enzymes. They lost their functionality, and even they damage the DNA also. So it can't replicate. So it will be dead. So this is the mechanism. So first it was well studied in a microbes how it inactivates. So there they observed that it can modify proteins, enzymes which are proteins, so which can help in inactivation of microbes. So why can't we apply it in food proteins? If it can modify proteins in the microbes, why can't we use in a normal protein? So this is where the concept of protein modification or biomolecules modification started. So they started applying for protein isolates to modify their functionality to use for various applications. So this is what I can take more, but if you have any specific questions, I'm happy to answer. Actually, one more thing I want to clarify. It's called cold plasma. Normally, what we call cold, if something is below 0 degrees, we call cold plasma. But it's not that cold. It is a relative term. Thermal plasma is around 1000. So when we compare with thermal plasma, the temperature here depends on the voltage. Maximum it reaches to 60 degrees, which is acceptable for the food modification. So it's a relative term when compared to hot plasma. This temperature is very low. So that's why scientists started calling it as a cold plasma. So maximum we can reach up to 60, not more than that. Maybe 30, 40. Now it's already cold outside, so it may be not that much. Actually, if you want to say exactly what's the temperature, there's a thermal camera you can take, and you take a picture when you are giving the treatment. So you can map that temperature distribution. So how much? It's mostly it's not more than 60. So it won't. So we are not going to lose any these nutritional properties, and we can able to inactivate microbes. No, it won't. Yes, we have to calculate.
Can we enhance the functional properties of some?
Yeah.
For example,
yes.
You said before, protein isolate.
Yes.
So if we apply in any food matrix or anything, if the protein isolates comes under cold plasma, whether the functional properties will be enhanced?
Yes. Okay. Actually, the one more disadvantage with cold plasma is it's a surface treatment. Let's say you have a protein bar, something very thick you want to modify. So kept in that, but it's a surface treatment. Its penetration depth is very less. So obviously you see the changes on the surface, not inside. So that's why it's mostly used as a surface disinfectant rather than a other modification technique. But if you want to modify protein, so you have to spread it in a thin layer, then you have to treat. So but when you take it to a commercial scale where we have to produce thousands of kgs of sample, then that may be issue. So that's why maybe you can go for a plasma activated water, so where we can use for the large scale production. And you have any doubt on cold plasma or anything, you can ask me.
So can this cold plasma can be used in this laminar airflow to disinfect this airflow chamber?
Yeah. Actually, that can be called as a indirect cold plasma treatment. There are two types of cold plasma treatment. One is direct, where you keep sample directly beneath that plasma generation pincer plasma electrode. But there is one other way, indirect way. So actually, I said most of the plasma species are short-lived, but there are some species which are long-lived. Let's say reactive nitrogen species. So actually, they can live up to maybe 30 minutes max. So you can maybe recirculate the plasma air to that chamber. Maybe you can disinfect it.
So after this technique, how do we detect all the microorganisms has been killed?
So you have to do plate counting, obviously. So you have to count the number of colonies alive. So let's say now you have a spirulina, cold plasma treated spirulina, and you have a non-treated one. So you count the number of colonies in that, and that the difference is the number of. So and one more difficulty is, so to call some technique as a sterilization technique, at least we need a 6-log reduction. So maybe cold plasma may not give that much reduction as a thermal techniques where we can completely reduce. So 1 log is 90%. So in order to get 6 log, I think we need to get 99.9949. That much is accepted at the industrial level to call a sterilization technique. So we can reduce it to some extent, which is safer level, but not as a sterilization technique. Any questions? And anything you want to know, not questions.
Okay, you haven't explained about the starches.
Yeah, starch. You want to know the starch, okay. Everybody, actually I'm working on starch only. My PhD topic is starch only, so I worked a lot on starch. So you know that starch is a semi-crystalline compound which consists of amylose and amylopectin. And each amylopectin, if you zoom it, if you go to micro scale, it's a glucose molecules. Molecules connected by a glycosidic bond. So what happens? So the functional properties of the starch. Let's say you want to prepare a gel. So native starch don't have as I said, don't have that much viscosity or a poor viscous properties. What you do? You keep a starch here, and what happens when this reactive species enters? So it breaks that glycosidic bond. So it breaks into basically depolymerizes the starch. So depolymerized first. So when you add it into water and when you cook it, so it's already depolymerized. So it easily mixes with the water and forms a thick, consistent gel. Where in the native form it forms a lump somewhere and somewhere less viscous liquid. So you don't have that consistency to prepare a gel. So
Solubility.
yeah. So it increases solubility. And actually the one more problem with cold plasma is it can reduce also, it can increase also. Let's say it has a amylose and amylopectin. Amylose is less amorphous, which is very less dense. So what happens when you're treating, to some extent it breaks only amylose and leaves amylopectin, which is very complex and dense. So obviously it reduces the solubility then, because all the soluble compounds are already broken down. So but when you treat up to some level, it increases. So the tailoring the results also very complex with cold plasma. Let's say you are treating, as I said, it retains some nutritional components to some extent only. There are few studies which show that excessive cold plasma treatment degraded not to great extent, but degraded the back to compounds to some extent. And one more disadvantage of cold plasma is, as I said, it has a react oxygen species. It oxidizes the lipids. Actually, samples which have high lipid content, let's say beef or pork, so it has high fat content. So obviously what happens, it reacts with the oxygen, react oxygen species, and oxidizes and gives us a little rancid taste, which we don't want. So but it has a lot of advantages. It has, I think, numerous I think n number of applications you can't even name it now. So it can be used for enhancing back to compound extraction, to enhance germination, to even degrade these pesticides, even modifying macromolecules. There are so many n number of applications, and you can maybe figure out one day a new application of cold plasma. Any question? Anything related? Cold plasma basics, if you want to know, you can ask anything.
Can we use liquid foods to treat
Little?
liquid foods like milk or something?
Here this is not actually intended for the liquid foods, but that we can use. That also the problem is the cold plasma where it generates, it creates a micro bubbles. The aperture is very small, so there are solids which are not soluble. It may stuck. So it's ideal to generate water there and mix it with something. And if you want to treat really liquid foods, then you have to purchase a setup, that plasma jet. So that may be ideal choice. This is mostly for yes.
So then what about the exposure? It may be different.
That actually it may be different for the, it completely depends on the sample type you are using, the thickness of the sample, and the ambient conditions that day. So we have to optimize it every time when you have to perform the analysis. That's it. And actually we have a fan over here, so if you want to make this reactive species generated really go into that, so you can use a fan also. Actually it's a continuous system, you can keep samples here, so you can turn on this, so conveyor belt, so you can adjust the conveyor belt speed in such a way that, so that meets the desired treatment time. And any doubts how it generates? Or, and one more thing, the one more disadvantage cold plasma is, so I increase up to 15. You can, as I keep on increasing the sound, you can voltage, you can hear that increase in sound. When you reach a certain level, you can see arcs. These are called dielectric arcs, which are not, which can damage the electrodes also. So when you are using high voltages and there are voltage fluctuations, so it creates arcs like flames, sparks it creates, and it will damage the electrodes. So you have to take care of that also. You can't go to higher voltages, and you can't, if you are keeping at low voltage, you won't get the desired properties. So you should figure out the optimum condition where you want to modify. And actually it's called dielectric barrier discharge. Along with the two electrodes, it has a dielectric barrier. Let's say we are sending we have two electrodes and we are sending a high voltage. What happens if there is no dielectric barrier? So it accumulates at the top electrodes, and the electricity won't able to pass to the gas. So it acts as an insulator and helps in creating a stable plasma. So that's why it's called a dielectric barrier. So normally this barrier made of ceramic or quartz. Most commonly quartz is used as a dielectric barrier, and always the ground electrode is grounded. So when something fails, so it transfers the high voltage to earth or somewhere, so that the person who is operating will be safe. And any questions if you want to?
After treating, the ROS will be present in this sample.
Uh-huh.
After treating, the reactive oxygen species is there inside the sample.
Yeah. Sometimes actually there is improvisation for this. Actually, that is called in-package cold plasma treatment. Maybe you can search. So what happens, even the packaging material we use can act as a dielectric barrier. So you have packed something in a package which is which keeps the generated species inside the package. You kept the package inside. So the package itself a barrier. You kept between the two electrodes, and it generates plasma inside the package. So after that also you can restrain that species inside that package. Even the sample contains for few minutes. Maybe reactive oxygen species don't have that much shelf life, a lifetime, but nitrogen species maybe for few minutes. And people till now don't know the adverse effects of consuming the plasma treated foods. That is also one limitation why plasma is not commercialized. What are the adverse effects on our own cells? Let's say you are consuming a food which still have a plasma species active. So what it would cause to us inside our GIT. So we don't know. So that's why more research has to be done on the animal models or somewhere where we can quantify plasma's negative effects on humans. Then one day we can see as a commercialized system.